Hello, I'm Anisa Abdi. I'm a sophomore from Stone Mountain, Georgia, and I'm studying biology in hopes of pursuing a career in forensic science after I graduate. So my topic is regarding these Namib desert beetles, who have the amazing ability to collect water from the fog that settles over the Namib desert, and they're able to use that for their drinking water in order to survive the harsh conditions of the desert. Okay, so now moving into the structure of this presentation, first I'll start with a little bit of discussing my inspiration for this presentation and my original mindset when I started this journey. Then I'll go into the background of these beetles and discuss their habitat and the actual process of water harvesting with the structure of these beetles. After that, I'll dive into the main focus of this presentation, which is the research paper where these scientists actually tested these beetles' abilities using 3 processes to determine their behavior and water collection efficiency. And the last bit of my presentation discusses the results they found and what that means for the field of study, before wrapping it up with the take-home message, including future implications for technology inspired by these beetle structures. So when thinking about what I wanted to discuss with this presentation in the early stages of my research, I was curious to learn about any kind of sort of phenomena of the world, particularly focusing on organisms of the world that are able to use special features of their bodies in order to perform certain tasks for survival. And that brought me to this field of science known as biomimicry or biomimetics, which is basically where science imitates nature in order to solve some kind of human problem. So this example shown on the slide is the creation of Velcro, which I did not know was originally based on these plants here on the left, known as burrs, that are also able to stick to various surfaces. And the scientist who designed Velcro, named George de Mestral, observed how these burrs were able to stick to clothing and even fur, and he used those observations as a starting point in designing the Velcro that is now used worldwide in various items. This story of the burrs was how I came upon my own topic with the Namib desert beetles, since their water-capturing dorsal structures are currently being used in modern research to design larger technologies to capture water from the air to be used by humans. So biomimicry was kind of the door that opened up and led me to my own topic. So getting into the background of these beetles, they live in the Namib Desert, which is a region that stretches across the southwestern coast of Africa, shown in this image in the bottom left corner. And there are approximately 3,500 species living in the desert, with a dozen species of desert beetles, and 4 of the main species I have listed here on the slide, with the corresponding images of each of these species in the top left corner. So all these species are able to water harvest, so I won't just be focusing on one species, since the main focus of this research is their behavior or structure, which doesn't vary that much according to one study which explored the efficiency of their water harvesting abilities. These beetles are able to survive in this desert even with only 0.5 to 2 inches of rainfall per year. The desert does, however, require a lot of fog, which is how these beetles obtain enough drinking water to survive. The fog is a result of the Benguela Current that flows along the west coast of southern Africa, where the Namib Desert is located, and it chills the air, causing a sort of weird area where the cool air rests at the bottom, close to the ground, and the hotter air rises, thus creating a cool cloud of fog that can get to be around 300 meters thick. Therefore, these beetles are able to use this cloud of fog that settles over the desert in order to obtain water for drinking. Now onto the actual mechanism of fog collection and how these beetles actually get water from the atmosphere. So shown in the image on the right, image B, you can see the basic structure of their dorsal surface, which has hydrophilic bumps and hydrophobic surroundings. So the water droplets from the air are attracted to these hydrophilic bumps and then roll down the troughs, or the spaces between the bumps that are the hydrophobic background, until finally the water slides all the way down the beetle's mouth and it pushes his mouth backward to drink the water, shown here in this illustration in image A, where the water droplets roll off to the mouth. And when they collect water, they get into a position known as fog basking, shown in this image on the left, where it basically just angles their back into the direction that the fog is traveling in order to capture the maximum amount of water from the air as possible. It's actually a fairly simple process once broken down like this, and one that has been observed by many researchers and many past experiments. However, there is still a lot that scientists don't know about these beetles, and much information they can still learn in order to help with future human creations of various water collecting machines. And that leads us into the main focus of this presentation, which is a paper titled Fog Basking Behavior and Water Collection Efficiency in Namib Desert Darkling Beetles. So these darkling beetles belong to a family of beetles called Tenebrionidae, which are classified by their dark color and small size. This paper's introduction explains that these beetles also possess what is known as elytra surfaces, which are the fused wings or hard forewings on their dorsal or back area that can either be on the smooth side with less ridges and bumps, or on the more bumpy side with more ridges and bumps. And this paper begins by introducing some of the previous work that has been done in this area studying these beetles. Studies have shown that it is essential for these beetles to be able to fog bask to survive, as beetles that are able to collect fog water have a much higher chance of living during periods of low rainfall than those beetles who are not able to fog bask. So what is already known in this field is how these beetles use their structures to collect water, that would be fog basking. However, what has not been previously explored by this prior research is the application of the mechanism of water collection by fog basking. It's argued in another paper that some beetles don't actually undergo this action of fog basking when they collect water from the fog, since some of their elytra surfaces do not contain the numerous bumps to collect water. Instead, there are some beetles with big bumps in their elytra, and some beetles with smaller grooves on their relatively smooth elytra. And as is shown in these images, the image at the top has a more smooth surface, and it's a species that doesn't have that much bumps, and then the beetle at the bottom has a lot more bumps and a more bumpy surface. Therefore, since the previous research did not really address the different elytra surfaces and just looked at if they were able to collect water or get into fog basking position, this paper tackles an analysis on the different level of efficiency presented with bumpy dorsal surfaces versus smooth dorsal surfaces. So the hypothesis is that elytra covered in bumps and valleys are better collectors of fog water than the smooth surface with grooves. And that leads us into the methods. So there are two main goals that the researchers wish to achieve with this experiment. First, to identify and describe a possible fog basking behavior in 4 desert beetles, and 2, to compare the water collecting efficiency of these two types of structurally different dorsal surfaces, that is, smooth versus bumpy surfaces. And through these two goals, they established 3 processes to analyze these beetles and obtain their data. First, they used scanning electron microscopy, or SEM, to see the texture of these beetles, if they were bumpy or smooth. And then they looked at the behavior, so if these beetles were able to fog bask or not. And if they were able to fog bask, they did a similar procedure before they looked at efficiency with the fog basking, where they changed the temperature and they also turned off the fog in their experiment to see what the triggering factor was and what caused the beetles to even get into the fog basking position to begin with. And then finally, they looked at the water collection efficiency and how much water each species was able to collect in a given time period. So the most collection of water would equate to the most efficient water collector. So the first process they completed was a closer look at the structure of these 4 beetle species using scanning electron microscopy. So what they did was they air dried these beetles and sputter coated them with palladium, and then they used Sudan 3 staining, which is a stain used to detect certain fats on various structures. So they use this staining process to test for wax that would be included in the hydrophobic properties of these beetles' elytra. So what they did was they placed the beetles in 50% alcohol for 3 minutes, and then placed them in the Sudan solution for another 3 minutes. Then the beetles were rinsed with 70% alcohol and then distilled water before finally being placed under the microscope for further analysis. And that process is what produced these figures shown here on the slide. So here we can see 4 panels, each one showing a darkling beetle species. Figure A is O. anglicularis, figure B is O. laviceps, C is S. chrysolepes, and D is P. quadripes. And the figures marked 1 show the beetle's dorsals as is seen by the naked eye, whereas the figures marked 2 shows the microscopic view of each beetle. The SEM process was only used to determine the structure of these beetles and to see if they followed the same hydrophilic or hydrophobic layout as depicted by previous studies. Of course, that wasn't the case, but the researchers still continued the study since their focus was on behavior anyway, not structure, and that led to their next process here, the fog basking simulation. So fog basking, as I mentioned previously, is the stance that these beetles adopt in order to situate their bodies to best capture the water from the fog. And the way that these researchers conducted this experiment was first by transporting the beetles from the Namib Desert to Oglethorpe University in Sweden, where they were conducting this research. The beetles were kept together in sand-filled boxes under the same living conditions, and during this transport, the beetles were given unlimited access to water, but a week after arriving in Sweden, the beetles were not given water for 2 weeks, and that is when the experiment was officially conducted. Next, they imitated the conditions of the Namib Desert during the fog period by using a fog-producing machine, and this fog-producing machine generated fog traveling less than 0.1 meters per second and produced 325 milliliters of fog water per hour. And they established this fog chamber where they placed the fog-making machine along with sand and the beetle into a box with a glass door, so they were able to observe and record the beetles. And the temperature was kept within a relevant range between 10 to 15 degrees Celsius, and it had a relative humidity above 97%. And the live beetles were studied one by one in a 17 by 17 centimeter square arena, and the sand was shaped into a small ridge oriented perpendicular to the direction of the fog, similar to how it would be in the Namib Desert. And then they recorded the time from which the beetle was placed in the chamber until it assumed a fog basking position. And this position was defined as the time at which the beetle had oriented itself with the back towards the fog and assumed a static position where it wasn't moving and it had its head tilted downwards for a minimum of 2 minutes. And the angle at which the beetle positioned itself was recorded with the camera they placed beside the fog chamber. And if no fog basking behavior was observed, or if the beetle did not position itself in the fog within the minimum 2 minutes, it was completely removed from this study and other behavioral tests looking at fog basking. And this elimination was crucial since in another set of experiments looking at fog basking, they altered the temperature in the fog chamber to either heat up to 20 to 30 degrees Celsius, or they completely removed the fog in order to determine what the triggering factor is behind the characteristic head-standing behavior of the fog-basking beetles. So they only included beetles that did perform this stance in the altering of the temperatures, since they wanted to specifically determine what caused these beetles to get into position. So that is, if it had something to do with temperature, or if it was just strictly dealing with the presence of fog. This procedure to determine the fog basking behavior of these beetles was to supplement their findings on this next part of the experiment, which was the quantification of water collection. So here they used all the beetles and not just the ones that performed in the fog basking procedure that they did previously. And for this next part, they actually had to kill the beetles and freeze them. And then they positioned the beetles on a stand at the angle at which they were positioned when they did the fog basking procedure. After that, they placed an Eppendorf tube under each beetle's head, so instead of the water that was collected on the back sliding into the mouth of the beetle, the water slid down the beetle and into the tube underneath. And the beetles were arranged in a random order in the fog chamber, and each still respected their previously determined stance angle. And after 2 hours, the water collecting efficiency of each beetle was established as the weight difference of the Eppendorf tube before and after the treatment. This experiment was repeated 5 times using a total of 20 beetles from each species. So finally, after all of these 3 processes, we can now discuss the results of each. So first, stepping back a little to the SEM procedure where they looked at the beetles under the microscope, they determined that O. ungicularis and O. laviceps had no bumps but had some ridges in their dorsals, and Escrocylipes and P. quadripes did have those hydrophilic bumps as shown in the standard structure previously. And in regards to the fog basking procedure, out of the 4 species, only 1 got into a stance where it was collecting water. That species was O. ungicularis, shown here in figure 4. And even though the other species were perfectly able to collect water, O. ungicularis was the only one that got into a stance that could probably be defined as fog basking. So continuing on to the water collection efficiency data, this chart here shows the amount of water collected by each species in their final process. The blue bars here show the total amount of water captured by each of the 4 species, and the red bars show the relative amount of water captured per dorsal surface area. The researchers found this value shown in the red bars by calculating the relative surface area of each beetle and then normalizing it to the largest beetle, which would be P. quadripes here on the right. And this produced a conversion factor to determine the relative amount of water captured per dorsal surface area. Even though the bar graph seems to show a great difference in the amount of water collected by each species, the researchers concluded that the 4 beetle species collected the same amount of water, over a 2-hour period in the fog chamber, meaning that there's no significant difference in the total amount of water that each of these beetles harvested. So the data in the blue, whereas the total amount of water captured, is not significantly different amongst each beetle species. However, they did determine that the collecting efficiency between these species was different. This efficiency found was based on the ratio of the size of the species to the amount of water collected. This particular data would be shown with the red bars. So the water collecting efficiency of the big species P. quadripes is significantly lower than that of the fog-basking O. anglicularis, which is here on the left. So with the data for O. anglicularis, it produced higher bars of data for collected water than did P. quadripes, even though P. quadripes is larger and should essentially be able to collect more water. Also, Esquicillipes here harvests almost twice as much water per surface area unit in comparison to P. quadripes, even though P. quadripes is larger and Esquicillipes is the smallest beetle among these 4 species. Therefore, there were significant differences in water collection based on size, but no significant difference in collection based on different dorsal structures. So with Esquicillipes being the smallest, and this data bar here, marked C, being the highest amount of water collected amongst all these 4 species, it was the one that essentially won this test. So it collected the most amount of water in the given 2-hour period. And this beetle species in particular, the Esquicillipes, did follow their hypothesis, which was that beetles that had more bumpy surfaces will be able to collect more water. However, P. quadripes, which also has bumps, did not follow their hypothesis because it produced the least amount of water, which completely counteracts or goes against their hypothesis, because this essentially should be, their bar should be much higher for P. quadripes because it has that bumpy surface. So if it was following their hypothesis, then it would be at least somewhere alongside Esquicillipes, if not more, since it's a bigger species. And also with O. anglicularis, it has a more smooth surface, so essentially it should have lower bars of data than these 2 Esquicillipes and P. quadripes, the ones that have a bumpy surface, since O. anglicularis has a smooth surface, so it should have less collection of water, but it goes against the hypothesis as well. So to sum up the results, their first main goal presented before conducting this experiment was to identify and describe a possible fog-basking behavior in 4 desert beetles. They did discover that O. anglicularis participated in fog-basking, whereas the rest of them didn't. And even with the temperature change, O. anglicularis was able to get into that ankle position, but it was only when the fog machine was turned off that it did not get into that position. So that shows that the presence of fog is a triggering factor of this head standing position, and fog is what causes them to get into that position. And with their second goal, they wanted to compare the water collecting efficiency of these 2 types of structurally different dorsal surfaces. So what they found with this data is that first, all the beetle species collected the same amount of water, regardless of structure, within the 2-hour period in the fog chamber. So more specifically, there was no significant difference in the total amount of water that they each collected. Also, despite O. anglicularis being the only one with fog basking abilities, it showed that the behavior was no advantage in collection of water. And instead, what happened was that size was the big indicator of how much water was collected. And surprisingly, the smaller sized species, particularly Esquicillipes and O. anglicularis, had a higher water collection to size ratio than the largest beetle, P. quadripes. Therefore, the smaller sized beetles are the most efficient water collectors. So overall, their experiment proved that their hypothesis was incorrect. Beetles with bumpy surfaces should have been the best collectors of water, but with the example of P. quadripes, the largest beetle that had a very bumpy surface, it collected the least amount of water when it should have essentially collected the most alongside Esquicillipes, the other beetle that had a bumpy dorsal structure. Instead, what they discovered was that size matters, and the smaller sized beetles were the best water collectors. And I did not expect these results personally. I thought that the largest beetle with bumps, which would be P. quadripes, would be the ones to collect the most water and be the most efficient. But the smallest beetles proved to be the best at collection. This result kind of reminded me of something I was learning in microbiology recently, in that there is actually a specific size of bacteria that is more preferred in nature and is essentially deemed as better or the better option, which also happens to be the smaller size over the larger size bacteria. And connecting that idea helped me to understand why these results in this experiment show that the smaller size is the best. This size is probably what is most suitable for the desert environment within which these beetles live, and therefore the smaller size is better off at collecting water and essentially surviving, since it can get more water than the larger species. And all this is to say that understanding the structure of these beetles and how they work is very important in modern science, especially in researching new technology for water harvesting. This image on the left comes from an experiment done in 2019 where the researchers used the beetle structures as inspiration to test out different models for possible structures that could be used in water collecting technology for human use. There are many other experiments being done using these beetles as a model to help understand the best way to collect water from the atmosphere, though researchers still have a lot to uncover about these creatures and the fascinating way they are able to survive such harsh conditions of the desert. And that is the end of my presentation. Thank you for listening.